Hi everyone, I am back with part 5 agricultural microbiology questions. Part 1 to part 4 link has been given in description, so please go through it. The first question is about chlamydia. Chlamydia are obligate intracellular parasite. Chlamydia trachomatis causes sexually transmitted disease. The G+C content of chlamydia is 41 to 44%. It's an energy parasite. Chlamydia has both elementary body and reticulate body. Elementary body is for infection and reticulate body is responsible for reproduction. Reticulate body can synthesize DNA, RNA, glycogen, lipids and proteins. And the second question is about fermentation. In Latin, the fermenter means to cause to rise. Louis Pasteur discovered the fermentation process in 1857. And the next question is, combined electrical and chemical potential differences make up the ATP are called proton motive force. Central role of proton motive force are bacterial flagellar rotation, active transport and conversion of ADP to ATP. And use of proton motive force for ATP synthesis is catalyzed by ATP synthase. In 1960, American scientist Ephraim Racker and co-workers isolated from mitochondria the enzyme F0, F1, ATPase, which we now call ATP synthase. And next, about subunit. Catalytic site of ATP synthase is beta subunit. ATP synthase consists of F0 and F1 complex. F0 consists of A and C subunit and F1 consists of B, 2 alpha subunit, 2 beta subunit, gamma, delta and epsilon. So here comes about glycolysis. Splitting of sugars is called glycolysis. It occurs both aerobic and anaerobic. It yields about 2 ATP and 2 NADH. Here it converts glucose to pyruvic acid. And next about pentose phosphate pathway. It is also called as phosphogluconate pathway and also called as shunt of glycolysis. Involve some reaction of glycolysis. So it is named as shunt of glycolysis. It's a glucose catabolism. So catabolism means breakdown of molecules. Here it obtains the energy from 5-carbon sugars. It generates NADPH, protons and ribose 5-phosphate. And next about ED pathway. It takes place in gram-negative bacteria, Pseudomonas, Rhizobium and Agrobacterium. It's also an glucose catabolism process. It generates 1 ATP, 1 NADPH and 1 NADH. It is present only in prokaryotes, not in eukaryotes. It found both aerobic and anaerobic prokaryotes. So, in fermentation, the organic compound acts as both electron donor and electron acceptor. And next about pyruvic acid. In aerobic respiration, the pyruvic acid enters into the Krebs cycle and supplies the energy to living cells. But in anaerobic respiration, the pyruvic acid ferments to produce lactic acid. So, it's a hub of carbohydrate fermentation. And next about glyoxylate cycle. It's an anabolism process and it occurs in peroxisomes. Here, acetate acts as the carbon source and specific enzyme involved are isocitrate lyase and malate synthase. 2-acetyl-CoA gets converted into succinate and 2-CoA. And next comes about Calvin cycle. In Calvin cycle, it's the reverse of pentose phosphate pathway. The chemical reaction performed by plants to fix the carbon from CO2 into 3-carbon sugars. Later, these 3 carbon compounds are converted into amino acids, nucleotides, and sugars. Finally, starch is produced. So, 3 stages in Calvin cycle are carbon fixation, reduction, and regeneration of ribulose. And next comes about gluconeogenesis. It occurs in liver and kidneys. Example, the synthesis of blood glucose from lactate in the liver. Gluconeogenesis occurs after 8 hours of fasting. It is important for maintaining blood glucose level during starvation. It is synthesized glucose from non-carbohydrate precursor. And next comes about hierarchical arrangement. Here, the domain is the highest taxonomic rank and species is the lowest. Domain, kingdom, phylum, class, order, family, genus and species. It's the arrangement. And the last question is about zoonotic disease, where the disease transmitted from animals to humans. So, the example for zoonotic diseases are anthrax from sheep, rabies from dog, West Nile virus from birds transmitted to humans. So, if you have any doubt, please ask into the comment section. For more videos, please like, share and subscribe our channel. Thanks for watching.